So we are in genetic mutation 17.6. And the learning goal will be to describe some ways in which DNA is altered to cause mutations. So first let us define what a mutation is. A mutation or change in the nucleotide sequence of DNA. So when we say nucleotide, what are we saying? In the beginning of this chapter, we had discussed what a nucleotide is. The nucleotide has three components. It has a nitrogen-containing base, which we have been discussing: adenine, guanine, cytosine, thymine. These are the nitrogen bases, attached to a sugar, attached to a phosphate. So all three together are going to, I'm sorry for the phone, okay. All three together, again I'm going to go back to talking about what a nucleotide is. A nucleotide is a combination of three units: the base unit with the nitrogen, the sugar unit, and the phosphate. So when there is a change in the nucleotide sequence of DNA, when the base component changes, there is a change in the sequence of the nucleotide. It alters the sequence of the amino acids. And because of the alteration in the sequence of amino acids, it affects the structure and function of a protein in a cell. Because amino acids are the components that create proteins. So when the sequence of amino acid changes due to mutation, it affects the structure and function of the protein. And these mutations may result from X-rays, overexposure to sun, chemicals called mutagens, some viruses. So in these mutations, if a mutation or change in the DNA nucleotide sequence occurs in a somatic cell, somatic cells are cells other than reproductive cells. Reproductive cells are called germ cells. So the somatic cell, if a mutation occurs in a somatic cell, a cell other than reproductive cell, like we just said, the altered DNA will be limited to that cell and its daughter cells. If the mutation here causes uncontrolled growth in these somatic cells, cancer could result. So mutation is the precursor to the uncontrolled growth in these somatic cells, and that could result to cancer. But if a mutation occurs in a germ cell, egg or sperm, then all the DNA produced in a new individual will contain the same genetic change. So now this has transferred to the offspring. So will contain the same genetic change. Now we're looking at the types of mutations. So types of mutations, the first, we can call that a substitution or a point mutation. In this mutation, there is a replacement of one of the bases in the template of a DNA with another base. Again, we said that the nucleotide has a base, a sugar, and a phosphate, so the base is replaced in the DNA template strand by another base. So if a substitution or point mutation changes the nucleotide, if we change the base, we are changing the composition of the nucleotide, a different amino acid may be inserted into the polypeptide. So if you remember a few units ago, we discussed how protein synthesis takes place. So the DNA template with the the information is transferred to the messenger RNA, to the transfer RNA. So if there is a substitution or point mutation, a different amino acid may be inserted into the polypeptide. If this produces no change in the amino acid sequence, it is called a silent mutation. What does this mean? If you go back to the codons that we were discussing, the triplets, the amino acids, a single amino acid has many codons. Sometimes we saw 3 to 4 codons. So even after replacing the base, it codes for the same amino acid. There has been a change in the base, but no change in the amino acid sequence. It's called a silent mutation because there are several codons that code for the same amino acid. In a frame shift mutation, there is the insertion or deletion of a single nucleotide into the sequence, resulting in a change to all subsequent codons. So codons are triplets. So we are coding 3, 3, 3, 3. So if you take one out, then you are completely changing the codons that follow. Or if you're adding a new nucleotide into the sequence, you're again changing the composition of the codons. So that is a frame shift mutation. So this is we are explaining what we just discussed. This is a normal DNA sequence, mRNA transcription translation. So let us see, this is the template strand. This is in protein synthesis. So we discussed this previously. So we see that this is the transcription message transferred onto mRNA. So thymine will code as adenine, adenine uracil, cytosine guanine. So this is the sequence on the messenger RNA, which is coding off the template of the DNA. So this message on the messenger RNA is translated by transfer RNA, and they bring the new amino acids and connect them in a sequence to set up the protein. So now the coding happens in triplets. This codes for methionine, that is the starting point. These three code for the amino acid. These three code for this amino. So this is how it proceeds till it comes here. This is the stop codon. So this is the amino acid sequence. Now this is a normal process of protein synthesis. Now if a point mutation or substitution has taken place, so we are replacing this by C. Okay? Substitution of C by T. So there was a cytosine, now there is a thymine. So when there is a cytosine, this becomes a guanine. Now there is a thymine, so it is an adenine. So the transcription has altered on the messenger RNA. So UAG now codes for this amino acid. UGG coded for glycine. UAG codes for serine. So now we have a new amino acid in the change in the amino acid sequence. So this is point mutation or substitution. Again, but like we said, that due to the change in the sequence, the protein structure and function changes. So that is mutation. Now let's look at frame shift mutation. The deletion of a base or addition of a base causes, so here we are deleting a base, deletion of a base. So now when you take this out, the triplets, we start here, triplet, triplet. These three were a triplet, now these three are a triplet. So we are changing the triplet sequence. So now the coding on the message, the transcription on the messenger RNA has changed, and the amino acids that are going to be brought up by transfer RNA are going to be different. So there is a change in the amino acid sequence, and this is frame shift mutation, shift in the frame. So now when a mutation happens, it causes a change in the amino acid sequence, which results in the structure of protein being altered, sometimes severely altered, losing its biological activity, because these are very specific to the sequence of amino acids. The proteins are very, very specific to the sequence of amino acids. So if there is a mutation or change in the sequence, it may severely alter causing loss of biological activity. When this condition is hereditary, it is called genetic disease. So here, DNA, due to these factors, may undergo alteration or mutation, creating a defective protein. When in the germ cells, it's a genetic disease. In somatic cells, it can lead to cancer. So mutation, the effects of mutation. So what is a genetic disease? A genetic disease is the result of a defective enzyme caused by mutation of its genetic code. Again, enzymes are proteins, and proteins are made up of amino acids. So amino acids are made up of the bases. So these are genetic diseases are a result of a defective enzyme caused by mutation in its genetic code. There are two examples here. Albinism results when the enzyme that converts tyrosine to melanin is defective. And in phenylketonuria, ketonuria, results when DNA cannot direct the synthesis of the enzyme phenylalanine hydroxylase required for the conversion of phenylalanine to tyrosine. So albinism is when we started this, we saw a peacock, just pure white, because it did not have the enzyme that converts this chemical to melanin, which is the pigment that gives color to the eyes, the skin, and all of that. So these are some genetic diseases, and this is in your book. So if you are not able to look at this slide, please open your book, and you have all these genetic diseases, table 17.8. So let's look at some of these. Cystic fibrosis is caused by mutation in the gene for a protein that regulates the production of stomach fluids and mucus. Cystic fibrosis is one of the most common inherited diseases in children, in which thick mucus secretions make breathing difficult and block pancreatic function. Then there is Down syndrome, there is muscular dystrophy, mutation in the X chromosome. This muscle destroying disease appears at 5. So these are some of the genetic diseases. Again, genetic diseases are a result of a defective enzyme caused by mutation in its genetic code. So please look at these diseases and their results. So let us look at the study check. Identify each type of mutation as a substitution or frame shift. Cytosine enters the DNA sequence. When you insert or delete a component, we call that the frame shift mutation, if you remember. One adenosine is removed from a DNA sequence. Again, you insert or remove, it is the frame shift. A base sequence of TGA and DNA changes to TAA. So this triplet change is called point mutation or substitution, yes, substitution or point mutation. So, okay.